So each Monday we have a 3-hour lecture, which is part of our course here, and that's the main lecture time that we get. And today's lecture was about the role of heterochromatin in aging, and we looked at its role in maintaining genomic stability. So basically, maintaining heterochromatin is really, really important. Again, heterochromatin is that really tightly packed DNA that prevents everything that's within to be expressed. And just a little bit of background, if you take a look at our chromosomes, those large structures of packed DNA, you would find that approximately, well, 30% of all of our DNA is heterochromatin, that is in the center of the chromosomes, and it's called the centromeric region, and it's very rich with repeated sequences. It's basically a really boring repeats, the satellite repeats, when the same sequence is just repeated over and over and over and over again, and it goes for, I don't know, thousands of kilobases. Well, yeah, really, really long repeats. And it also contains retrotransposomes, those analogs of viruses. Those sequences can actually self-replicate and integrate themselves into other parts of the genome. They can jump to different chromosomes even. So what's happening in the centromeric region, it needs to be stable. It should not be too active, and in fact in young cells that's the case. Only over time, with aging, the heterochromatin organization is kind of degraded, and we see a loss of heterochromatin over time. And so one of the proteins that help maintain the heterochromatin status, this real tight packaging, is called heterochromatin protein 1, HP1 for short. So there was the study, and people found that when you overexpress this HP1 protein, you can get about 20% of life extension, and that's maximum life extension in flies. And this is a really, really neat result. And in fact, if you overexpress it even more, the flies will not survive, but a slight overexpression, it means a slight increase in protein production leads to very nice life extension effect. So I'm going to talk about why this may be happening, why it has such an impact on the aging mechanisms a little bit later.